Hello everyone, welcome back to my channel. This is lecture 2 of chapter Structure of Chromosome, Cell Cycle and Cell Division. Okay, and in this lecture we are going to do what? We are going to do revision, study new topics like nucleosomes, then structure of DNA, formation of new DNA, what are genes, and in the end we will be discussing about our study plans, like how you can manage your studies and the different subjects and which subjects require daily practice. So we will be discussing about everything, so stay tuned to the video. Okay, so let's begin. Our first lecture we studied about chromosomes, so let's revise chromosomes. Okay, so this word chroma means color, and this word zoma means what? They means body. So chromosomes are colored bodies. They take up stains. Now, we also studied that chromosomes are highly condensed coiled chromatin fibers. Suppose this is our chromatin fiber. Okay? They condensed and they form chromosomes. So, chromosomes are highly condensed, coiled, chromatin fibers. Okay, now this chromosome has two chromatids. They are also known as sister chromatids. This is one chromatid and this is another chromatid. In center they are joined by centromere. This centromere acts as a point of attachment of both the chromatids, and they were also acting as a point of attachment for the spindle fibers during cell division. Okay, now if we talk about the discovery of chromosomes in 1882, who discovered? Who? Fleming discovered. Fleming discovered this colored body, okay, which is known as chromosomes. Now, okay, this chromatin, it also consists of two things. One is DNA. Full form is deoxyribonucleic acid. Which is found 40%. Okay, this is 40%. DNA is 40%. Then another component is histone proteins. These histone proteins occur in the form of octamer. There are 8 subunits which form this histone proteins. Okay, and this histone protein, they are found 60% of the total content. Okay, I hope everything is being revised clearly. So let's begin with our first topic, which is nucleosome. nucleosomes. I am drawing DNA. This is DNA. Now, next I am drawing is histone proteins. Histone proteins are octamer. 8 subunit of histone proteins are found. So this is first octamer, this is second, this is third, this is fourth, fifth, sixth, seventh and eighth. They are behind this. This is the first 4 octamer and behind the next 4 octamer are present. Okay, now we are wrapping this DNA around this histone protein. So what we get? See. All are octamer. This is one set of histone octamer, this is another, and this I am drawing another. Now, this DNA is being wrapped around this histone like this. So, whatever structure we are getting after this DNA is being wrapped around this histone octamer is called nucleosome. So, here we can see 3 nucleosomes. This is one nucleosome, this is another nucleosome and this is another nucleosome. So, what is nucleosome? Means histone, and on this histone the DNA which is wrapped. So, both are together known as nucleosomes. So, here these 3, all these 3 things are called nucleosomes. So, we may write nucleosomes as DNA wrapped around the core of the core of 8 histone molecules. Okay, now this is nucleosome. Now if we wrap this whole structure, we coil and supercoil it. Means we fold and fold and fold. So what we get? We will get chromatin fibers. Okay, after coiling this, we are getting chromatin fibers. See how. We have coiled it. This is chromatin fiber. Now, again we are coiling it further. Means again we are condensing it. We are condensing this chromatin and we will get what? We will get chromosomes. This is chromatin which we are condensing again and we are getting chromosomes. Okay. I have explained it very simply. Okay. First DNA, this DNA is combining with histone and it is forming nucleosomes. Then from nucleosomes, these nucleosomes are coiling and supercoiling and they are forming chromatin, and from chromatin they are condensing and forming chromosomes. Okay. You may note this point that in human a single chromosome may have millions of nucleosomes. Okay, note this point. Write this point in your notebook wherever you are making notes, that a single chromosome may have millions of nucleosomes. Okay, now let's move to the next topic. structure of DNA. So the shape of DNA was studied by a very brilliant scientist named Rosalind Franklin. In 1953. Okay, studied shape of DNA. Okay, she was a very brilliant scientist of Britain. Okay, she did her PhD in chemistry, and with the help of X-ray machine, she observed this structure of DNA. And the picture which she took of this structure was called picture 51, because she took that picture in year 51, but according to your book it's mentioned 1953, that's why I've written 1953. Okay, then what happened? Her partner, he took its image. Okay, he took the image and he went without telling Rosalind Franklin. He went to Watson and Crick, and with them they all three combined and they published the further structure of DNA, which is known as double helix model. So double helix model, which is being proposed by Watson and Crick in the same year 1953. Okay, but the actual work was of Rosalind Franklin. She was the first one to observe the structure of DNA. Okay, but in 1958 she died and the Nobel Prize was given to them. Okay, not her, because a Nobel Prize is never given to a dead person. But you know, the main contribution was of her. So in honor of her, I am telling this story. Let's see what DNA is. Okay. So this DNA is a single large molecule. Means it is a macromolecule. Why macromolecule? This macro means large. So it is a large molecule. Now, this DNA consists of two complementary strands which are bound around each other. I will be explaining. Okay. So it consists of two complementary strands wound around each other. See how I am explaining this point. Suppose this is one strand, this is another strand, this is one strand, this is another strand. Now, so two strands which are complementary. Complementary means if one side is 5 dash to 3 dash, we are naming it 5 dash to 3 dash, other strand will be 3 dash to 5 dash, means they are complementary. In front of 5 dash we will have only 3 dash, not 5 dash. Okay, like the magnet, north always attracts south. Okay, north will always repel north. So similarly, in opposite to 5 prime end we will always have 3 prime end. Okay, now, so they are complementary, wound around each other. They are wound like this. Okay, 5 dash, 3 dash, 3 dash and 5 dash. Okay, now note this point. This each strand consists of repeating units called nucleotides. Each strand, write this point, note this point. Each strand consists of repeating units called nucleotides. Now we are going to study about nucleotides. Okay? I'm changing the color of the pen. Now, see. Nucleotide. It consists of 3 components. One is phosphate, another is pentose sugar. Pentose means 5 carbon sugar, okay? And the third is nitrogenous base. Nitrogenous bases. Examples of nitrogenous bases are, okay, their structure have nitrogen, that's why they are called as nitrogenous bases. Examples are adenine, guanine, cytosine and thymine. Now, let's see how they are attached to each other. See, this is a phosphate. This phosphate is attached with pentose sugar. Pentose sugar is 5 carbon sugar. And with this sugar, we have our nitrogenous bases attached. So this is our basis. Okay. Now, this is one nucleotide. Okay. Now, this one nucleotide will be attached to the another nucleotide. Suppose this is one strand, this is another strand. So, this one strand nucleotide, with the help of this nitrogenous base, it is attached to the other nucleotide, okay, and that another nucleotide forms the another strand, okay, and there is hydrogen bond in between, which is joining both the bases. So I am again drawing another nucleotide also. So in between we have hydrogen bond. This is hydrogen bond. Okay, now I am changing the color. Again we have base. With base we have our pentose sugar. And with this sugar we have our phosphate. Okay. Similarly, this is another nucleotide. And this goes on because this whole strand is made up of different nucleotides. So this goes on. Similarly, below this nucleotide we will have another nucleotide, then another nucleotide. Similarly, below this also we will have another nucleotide, and they both will be joined. Yeah, the base of one will join the base of the next strand, okay, with the help of hydrogen bond. Now we will be studying about this basis, and the bases which will join each other, means the bases which are complementary to each other. Let's study that thing. So This base adenine, it is A, it is denoted by A. Guanine is denoted by G. Cytosine is denoted by C, and thymine is denoted by T. So always remember this: adenine pairs with thymine with the help of two hydrogen bonds. Here two hydrogen bonds. Okay, this is one hydrogen bond and this is the another hydrogen bond. And this guanine always pairs with cytosine with the help of three hydrogen bonds. Okay? Just remember this. Always remember this. When I was in class 10th, I learned this point because my pet name starts with T. So I always want to pair with A grade. So I learned that way. So you may find out any trick to learn this thing, or you may remember this G pairs with C because they both have similar alphabetical shape, like G or C you will make like this only. So they will always pair together, means G will always pair with C and C will always pair with G with the help of three hydrogen bonds. So if you remember one, automatically you will remember the second one also. So I hope it is clear. Okay. Before understanding like how DNA is formed, let's understand that why we need new DNA. Okay. We know that within second our body produces so many cells, like from one cell, two new cells are formed, from this further two new cells are formed. So whatever content was there must be getting duplicated, which must be being transferred to the other cells. Suppose if DNA is present, it must be getting duplicated, which must be getting passed on to the next cells. Then again here also duplication will be taking, which again must be passed on to the next cell. So I hope you understand the reason why we are forming new DNA, like why our body is producing new DNA. Now let's understand how this process occurs. Okay, now so this formation of new DNA, it occurs in the interphase or the resting phase of the cell, interphase stage of the cell. Means when the cell is preparing for division. Now, and this process, this formation of new DNA is called as replication. It is called as replication. DNA. Okay, now see this in interphase. See, this is our chromosome, because this chromosome, it is formed due to the condensation of chromatin fibers only. So our DNA is all over it. Okay, this is the DNA. Okay, now so during the interphase stage, what is happening? This DNA is getting duplicated. How? This DNA, both the strands of this DNA open. Wait, I am drawing. Suppose it is opening. From upper side it has opened and from lower side it is still wound. Okay, so from the upper side it has opened. So this is acting as the parent strand. Both the strands are acting as parent strand. Now the new DNA will formed on the strand which has opened. So here new DNA is being formed. Okay, So over time, the whole strand of DNA will open and new DNA will be formed over the whole strand. So now, this also happens that initially our chromosome was looking like this. Now, our chromosome has two chromatids. Initially only one chromatid was present, now we have two chromatids. Because our amount of DNA has also doubled. So, this is the new DNA which has formed. Okay. Initially we were having just this one DNA. Now we have two DNA. I hope it is clear. Our DNA has doubled. Okay. I hope you understood this formation of new DNA, like how it is formed. The parent strand separates and over that parent strand our new DNA, new dotted DNA is being formed. Okay, and that process is called replication and it is occurring in the interphase stage of the cell. Let's understand what are genes. So genes are specific sequences. Okay, genes are specific sequences of nucleotides present on chromosome they are present on chromosome that encode for a particular protein. I will be explaining, okay? That encode for a particular protein. Okay? This particular protein, it is associated with a specific feature which tells that what kind of hair color we will have, what kind of eye color we will have, how tall we will grow. Okay? Everything is being decided by these genes. Okay? So remember, they are just the sequences of nucleotide which we have studied. Okay? Like we studied about the nucleotide phosphate, pentose, sugar, and here was our base. So these are just the sequences of nucleotide which are present on the chromosomes and which are encoding for a particular protein, and that particular protein is giving a specific feature to our body, and that specific feature is identifying our specific characters. Okay, I hope these genes are very simply clear. Okay, now let's discuss about the study plans. Okay, 3 things I am going to tell you. First is keep a track of your studies. I will be telling how to do this. Okay, keep track of your studies. Okay, on a piece of paper or in your notebook. Okay, just write down the date. Okay, and write down all the work that you have to do for this particular day. Like the work, like you have to solve 5 questions, write down 5 questions of maths. If you have to do one exercise, write down one exercise of this chapter of this subject. Okay, now you may write down, and before going to sleep, just put a tick mark in front of that. It's okay if you are not able to complete everything. It happens. Okay, so no need to be worried. Try to do it the next day. Okay, this is the first thing. Okay, you will remember, you will be in a very good position after 1 month. Just try this thing. Okay, believe me, I have myself done this thing. That's why I'm asking you to do the same thing. Now, the second thing is take help. Take help from your teachers or friends. Okay, for subjects for which you don't have any idea like how to deal with subject, especially it happens in case of English language and Hindi. So you may take help from your teacher. Ask your teacher, teacher please help me to understand this subjects, or how shall I study this subjects, or how much time shall I give this subject. You may ask your teacher or your friends. Okay, I remember that when I was in class 10th, I took help from my friend. She was the topper of our class, so she was the one who told me to write a letter once a week, an essay once a week, and get it checked by our ma'am, or I may give it to her only to check it. So from there I started working on my English grammar. So you may also try this thing. Okay, now now the third thing is, which is very important, please include maths, English and Hindi in your daily routine schedule. Which subjects? English, maths, Hindi. Okay, English means grammar portion and Hindi also grammar portion. Okay, in English you may solve the transformation sentences, or you may practice different things. Okay, and Hindi also. I remember that when I was in class 10th, newspaper used to come to our home, and from that newspaper there was a story section. I used to read that story section, and you know what, in the final boards exam, the topics which came in exam were very difficult. So I had no option, but I had to choose that picture comprehension one. And from so for that picture comprehension, I wrote a story which I had already read in newspaper. So it was very helpful for me. And because of that, I really scored good marks in Hindi in my board. So you may also do this. Okay, and maths is also very important. Okay, no matter you're solving just one question, two question, just don't break the consistency. Try solving maths on a daily basis. And one thing I want to tell is, apart from this studies, please take care of yourself. Your health is more important than all these things. If you will be happy, you will be fine, you will be able to do everything. Okay, so just calm down, just believe in yourself, just believe that you can do it, you will be able to do it. Okay, I know it's really difficult time is just passing and passing, but it's okay, it happens with everyone. So just calm down, relax, and just focus on your target. Okay, this thing you may do it on a daily basis. And physics, chemistry, biology, you may include, or you may give three days in a week. So just try it out and just take care of yourself. Thank you so much for watching this lecture. See you in the next video. Thank you so much.